Hi everyone, this is Kalyan Kumar this side and welcome back to Chemistry Universe. This is the last video on carbohydrates. After this we will be doing proteins and we are doing biomolecules if you remember. And this is the first part of biomolecules, carbohydrate. That itself contains 12 videos and this is a very, it's going to be a longish video, but mainly because I'm going to take you to the lab and show you the tests. So this is about test for carbohydrates and there are various important tests. Let's look at each one of them. All right, so this first one is the heating test. And this what happens is when heated in a dry test tube, the carbohydrate melts, turns brown and finally black, giving a characteristic smell of burning sugar. So this is a heating test and it's a very simple test that you can do. You can perform even in your home. You can take table sugar and in a spoon and heat it and check for yourself. But I'm going to show that how it is done and what it looks like. So let's go and have a look at it. Welcome back. So that was the heating test. The next one that we're going to look is the sulfuric acid test. So this is the sulfuric acid test. And here when warmed with a little concentrated sulfuric acid, it leaves a charred residue of carbon. So it's a very simple test. And you will find the black carbon residue left. So let's go to the lab and have a look. So welcome back and now let's look at the next one and that is called the Molisch test.
Molisch test. Now there are several tests available for detecting and differentiating reducing sugars from non-reducing sugars. That is the Tollens, Fehling's, Benedict's and Barfoot's test. These are only given by reducing sugars. All monosaccharides, that is aldoses and ketoses, give all these tests, though these tests are primarily indicative of aldehyde group. Now, as far as ketoses are concerned, they are also primary alpha hydroxyketones, which can tautomerize to the aldehyde form in these tests. The monosaccharide aldoses and ketoses can be distinguished among themselves by the Schiff's test, as they are only meant for aldehyde group. Unlike these tests, the Molisch test is a test for carbohydrates in general. Now, all carbohydrates, including monosaccharides, disaccharides, and polysaccharides, except trioses and tetroses, show positive Molisch tests. Now, ideally, this test is only given by monosaccharides. But since you have disaccharides, oligosaccharides, and polysaccharides, in the hemiacetal form, they will first hydrolyze to the monosaccharide units, and that monosaccharide unit would give this test. Due to this, the monosaccharides tend to give this test faster. Now, the reason why triosis and tetrosis do not give this test can be understood by the lack of formation of furfuryl, which is a cyclic 5-membered ring compound formed when monosaccharides react with acid. So let's first of all understand what is this furfuryl. So whenever a monosaccharide reacts with concentrated HCl or sulfuric acid, and if it is a 6-carbon compound or a 5-carbon compound, it tends to form furfuryls. For example, let's take the example of glucose. When we treat it with an acid, the OH on the 3rd carbon, that is this carbon, would get protonated, and then elimination will take place, and that will lead to an enol. This enol will tautomerize and give a ketone. Then the OH on the 4th carbon would get protonated, and this is called the 3-deoxyglucosone, and this gets protonated, and again elimination occurs between this hydrogen and this water molecule, and you tend to get another enol. Now, before this even tautomerizes, the lone pair on the ketone O takes the H plus and forms a carbocation, which is then attacked by the lone pair of this oxygen. So this attacks this carbon, and you get the cyclic compound. Now this shows intramolecular proton exchange. The H goes from one oxygen to another, and then you have another dehydration happening, giving you this compound. Now this is called 5-hydroxymethyl furfuryl. This is carbon 1, this is 2, this is 3, this is 4, and this is 5. On the 5th carbon, you have the hydroxymethyl group. So this is called 5-hydroxymethyl furfuryl. This part is furfuryl. And on the 5th carbon, you have hydroxymethyl group. Now, obviously, we get 5-hydroxymethyl furfuryl only because we have 6 carbons. If we had only 5, then this would not exist. And therefore, that would only give you a furfuryl. So, pentoses will not have the CH2OH group, and that product is called furfuryl. That is this one. Now, it is this furfuryl that reacts with the Molisch reagent. Now, since the formation of the cyclic compound requires at least 5 carbon atoms, tetrosis and triosis cannot give this test. So now let's understand what the test is. First, let's look at the chemical reactions. I'm taking the example of 5-hydroxymethyl furfuryl. This is reacting with Molisch reagent, which is basically an alcoholic solution of alpha naphthol. In the presence of acid, you get an adduct like this, which is a purple colored dye. The same would happen even if you were to take a pentose, which would only give you a furfuryl, and yet it reacts with alpha naphthol, and this also is a purple colored dye. Now, the alcoholic solution of alpha naphthol, that is called the Molisch reagent, is added to aqueous glucose solution, and 1 ml of concentrated sulfuric acid is added carefully along the sides of the test tube. The formation of a violet ring at the junction of the two liquids confirms the presence of carbohydrate. Now let's look at what the test is in the form of a demonstration. Now in this demonstration, I'll show positive Molisch test using a few carbohydrate solutions as test samples. To perform the test, we'll need the following: the Molisch reagent, concentrated sulfuric acid, solutions each of the following: fructose as representative of ketose, monosaccharide, glucose as a representative of aldose, sucrose as a representative for disaccharides, and starch as a representative sample for oligo and polysaccharides, and distilled water as the control sample. 5 clean, dry, identical test tubes, a test tube holder, a test tube stand, 7 graduated droppers or pipettes of 1 or 2 ml capacities. Now take the 5 clean, dry, appropriately labeled test tubes in the test tube stand. Using 5 different pipettes, add 2 ml each of the carbohydrate sample and the control sample to the 5 tubes. The tube containing distilled water will be the negative control for this experiment. Now, using a different dropper or pipette, add 2 to 3 drops each of Molisch reagent to all the 5 tubes. Mix the tube contents by gently swirling. Using a different dropper or pipette, gently and slowly add 1 ml of concentrated sulfuric acid along the sides of each test tube so that the 2 distinct layers are formed in each tube. Now observe any color change at the junction of the 2 layers in all the tubes. Appearance of a purple ring at the junction of the 2 liquids indicates the presence of carbohydrates. As expected, this purple ring is formed in all 4 test tubes containing the test samples because they are all carbohydrates with 5 or more than 5 carbon atoms in their constituent monosaccharide units. The negative control tube, as expected, shows negative reaction as well. So there are certain limitations to this test. The limitations are that the Molisch test yields no results for triose and tetrose, so you cannot identify them. As a result, even if the sample contains any triose or tetrose carbohydrate, the sample will still yield negative results. So a negative result does not necessarily mean that there is no carbohydrate. Moreover, it may give positive results in the presence of any furfuryl type chemicals like citric and lactic acid. It will also give positive result on nucleic acids and glycoproteins. That is the Molisch test.
So welcome back. That was the Molisch test. And with that, we come to the end of this particular video, the test. This is part 12. And we saw all these tests. I thought it would be easier to remember these tests if we could show that actually happening in the lab. And so I hope this video helps you in understanding these tests. And that concludes carbohydrates for us. And the next one that we're going to begin are the proteins and amino acids. So that would be the next biomolecule that we'll take up. So with that, this is Kalyan signing off. Have a great and wonderful day. Hope you were able to understand carbohydrates in detail. And thank you for watching this.